let's talk about one of the most powerful tools in all of modern biology. And what's wild is that we didn't invent it. We discovered it right in the middle of an ancient microscopic war. We are talking about restriction enzymes. You can think of them as DNA's original molecular scissors. So think about this for a second. It really comes down to a fundamental question of survival. You've got a virus, literally thousands of times smaller than a grain of sand, and it's attacking a single bacterium. How on earth does that bacterium fight back? Well, the answer is just so elegant, and it's completely changed our world. Okay, so here's the scene. Bacteria are pretty much under constant assault from these viruses called bacteriophages. You can picture these phages as tiny biological needles. They lock on to the bacterium's surface, and then they inject their own viral DNA inside. Their goal is simple: hijack the cell and turn it into a mindless virus-making factory. But, bacteria are not going down without a fight. They are not helpless. Over millions and millions of years, they have evolved this absolutely brilliant defense system. They produce a special kind of enzyme that's basically a highly specialized pair of scissors. It's designed to do one thing: find that invading viral DNA and just chop it up into useless little pieces. And this brings us right to the hero of our story, the restriction enzyme. Its one and only job is to find and cut foreign DNA. The name itself is actually pretty cool. Early scientists noticed that these enzymes restricted the growth of viruses trying to infect the bacteria. So, restriction enzyme, it does exactly what it says on the tin. Now, here's what's truly amazing. The precision here is just off the charts. These enzymes don't just snip DNA randomly. Oh no, they are programmed to find a very specific sequence of DNA letters, what we call a restriction site. For a famous enzyme like EcoWAR1, that target is always GAATTC. If even a single one of those letters is wrong, the enzyme just ignores it and keeps on moving. To really get how they find these sites, we have to talk about something you probably learned about in English class: palindromes. A word like madam that reads the same forwards and backwards. It seems simple, but this concept is the absolute key to how these enzymes lock on to their target. So how does that work for DNA? Well, it's a little more complex because, as you know, DNA is double-stranded. A palindromic sequence here means that the letters on the top strand, read from left to right, perfectly match the letters on the bottom strand, read from right to left. This creates this perfect molecular symmetry that the enzyme is physically shaped to recognize. It's like a perfect handshake. Okay, but this brings up a massive logical problem, doesn't it? There's a huge, potentially fatal flaw in this whole defense system. I mean, really think about it. If the enzyme is just looking for a specific sequence, what happens when that same sequence shows up in the bacterium's own DNA? And statistically, it almost certainly will. What stops these molecular scissors from committing cellular suicide and just shredding their own genetic code? Well, the answer is just incredibly clever. The bacterium evolved a way to put a disguise on its own DNA, basically making it invisible to its own security system. This whole setup is called a restriction modification system. Think of it like a two-part security team. You've got the restriction enzyme, the scissors, and then you've got its partner, an enzyme called a methylase. The methylase's job is to go around and add a tiny little chemical tag, a methyl group, to the bacterium's own DNA, right at the very spots its partner is programmed to cut. And the result is a beautifully simple system. When that viral DNA gets injected, it's naked. It doesn't have those chemical tags. So the enzyme sees the restriction site and snip, it cuts it. But when that same enzyme comes across the same site on the host's own DNA, it sees that protective methyl tag. That tag is a clear signal: hey, this is one of us, do not cut. The host DNA is ignored, and it stays perfectly safe. So, as you can probably guess, this incredible defense mechanism didn't stay a secret in the microbial world forever. Once scientists figured out what was going on, they realized they had just stumbled upon a revolutionary tool that could change biology for good. As researchers started digging into them, they found there are actually a few different types of these enzymes. And they learned pretty quickly that only one specific type had the kind of predictability and precision they needed for the super delicate work of engineering genes. It turns out there are three main types. Type I and type 3 are, let's just say they're a bit wild. They find their specific site, but then they cut the DNA randomly, somewhere else down the line. That's no good if you need to be precise. But type 2 enzymes, they are the absolute gold standard. They find their site and they cut right there, inside that sequence, every single time. That reliable precision is exactly what scientists were looking for. And that's why these super useful enzymes all get specific names, like the famous EcoR1. It's not just a random name, it's like a code that tells you its whole origin story. E for the genus, Escherichia. Co for the species, E. coli. R for the particular strain it came from, and the Roman numeral I, because it was the very first one found in that strain. Pretty neat, right? Now here's where the real magic happens for us in the lab. Because these type 2 enzymes often make a staggered cut across the two DNA strands. They leave these little single-stranded overhangs. We call them sticky ends. You can literally think of them like molecular Velcro. They have a few unpaired letters that are actively searching for a matching sequence to stick to. And this right here is the fundamental principle that blew the doors wide open for all of genetic engineering. If you take two totally different pieces of DNA, say one from a person and one from a bacterium, and you cut them both with the exact same restriction enzyme, they will both end up with perfectly matching, complementary, sticky ends. They're literally made for each other.
So how does this actually work? It's really like a biological cut and paste. First, you take your enzyme and you use it to cut open a small ring of bacterial DNA called a plasmid. Then, you use that same enzyme to cut out your target gene. Because you use the same enzyme, they now have those matching sticky ends, and they just naturally find each other and snap together. Then, a final enzyme called DNA ligase comes in and acts like molecular glue, sealing everything up tight.
What you've just made is a recombinant plasmid, a brand new hybrid piece of DNA. And here's the payoff. When you put that engineered plasmid back into bacteria, those bacteria will read that new gene you just inserted and start pumping out whatever protein it codes for. Things we desperately need, like human insulin, a discovery that has saved countless lives. So there you have it. A simple defense mechanism from a bacterium, a pair of molecular scissors forged in an ancient microscopic war, became the key that unlocked the entire age of genetic engineering. It completely changed medicine and science forever. And it just leaves you wondering, doesn't it? What other doors are these incredible tools from nature going to help us open next?